Hey guys, it's part 3 of the genetics practice questions from last week. I'm going to start with number 10. Body cells include nerve cells and muscle cells, two different kinds of cells. Each makes a number of different proteins. For example, nerve cells make cholinesterase, which is an enzyme with the ACE, and muscle cells make myosin. Which statement best compares the DNA normally found in these two types of cells in an individual? Okay, so how can we compare these two types of cells that make these two different types of proteins? The two cells have identical DNA. These two say identical DNA, these two say different DNA, okay? Every cell in your body, other than your gametes, have identical DNA. Okay, so they're not going to be different. The only cells in your body that have different DNA from each other are the gametes, are the sex cells. So these two are totally out. Yes, a small mutation could happen, but in general, they're identical. They start out as being identical. Every cell in your body has the identical chromosomes, same DNA. So if they have the same DNA, how are they going to be able to make these different proteins, right? Okay, they use the same section of the DNA to make the proteins. Well, if they use the same section of the DNA, then they would be making the same protein. So they must be using different parts, different sections of the DNA. So they have the same DNA, but they're not both using the same parts of the DNA. They're using different parts. So the answer is B. Number 11. This you already did last week. This was part of the text reading, so this should have been easy for you. All right, a sample of DNA from human skin cells contains 32% cytosine bases. So if we know there's a 100%, right, that's what we're going to get to. That's the total amount. And 32% is cytosine. What does cytosine bond to? Cytosine bonds to guanine. So if there's 32% cytosine, that means there has to be 32% guanine because they're bonding together. All right, so that gives us 64. All right, so what's left from that, 100 minus 64, is 36. So then you're saying, okay, well, whatever's left has to equal 36. We have adenine and we have thymine. Okay, so basically you just divided 36 in half. So there's going to be 18 adenines and 18 thymines, 18%. Which is going to equal 36, okay? They're in equal amounts. They're in equal amounts. Cytosine and guanine are going to be in equal amounts, and adenine and thymine are going to be in equal amounts, okay? So how much thymine? There's going to be 18%. All right, let's go down to number 12. Base your answer to the following question on the diagram below and on your knowledge of biology. The diagram shows a small segment of DNA taken from a gene before and after it's copied. Changes made during the copying process are represented by the star. Okay, so basically we know this is DNA, and we know this is DNA because there's thymines in it. Only DNA has thymines in it. Well, this is not RNA. It's saying it's being copied, so we're just replicating. We're replicating before cell division. So here's before replication, and here's after replication. Right? It says before and after it is copied. So mRNA, when you make mRNA, you're not copying the DNA. Okay, you're not really making more of it. You're making mRNA, you're making a different molecule. Here we're making more DNA. So what are the errors indicated by this star? What could that affect? So there's an error here. So we have a G here instead of what's supposed to be a C. So that's one little mutation. And then we have a C here where there's supposed to be a G. What could that affect? We don't know if it actually is going to affect anything. It might be a silent mutation, meaning that it will code for the same amino acid anyway. And in that case, no problem. That's called a silent mutation. But maybe it does code for a different amino acid. And if it does, then you're going to have a problem with the protein. Okay? So what could this affect? Altering the number of chromosomes? No. Converting the original cell into a different type of cell? No. Converting sugar molecules into molecules of protein? No. Changing the sequence of amino acids. It's all about amino acids. That could change the sequence of amino acids. All right. We're gonna move on to these because you also had these to do last week. And a bunch of people were asking me how to do this. But if you watch the video, you shouldn't have a problem with this. If you don't know how to do this, you need to go back and watch the video. Okay, that means you didn't really watch. That's what that tells me. If you're asking me how to do this, that tells me you didn't really watch the video and you didn't go back to watch it again when you knew that you didn't understand it. Okay, I'll do it one more time, that's it. Okay, transcribe the mRNA. So now this is not making more DNA. Now the DNA is opening up to code for making a protein in the cell at the ribosome. So the first part that's gonna happen, this part right here, is gonna happen in the nucleus. Okay. Everything else after that is going to happen at the ribosome. Okay, first, that's important. So, how does it get transcribed? Well, we said that thymine in DNA bonds with adenine. Okay? If you see an adenine, it would normally bond with a thymine, but guess what? There's no thymine in RNA. There's a uracil instead. If we see a C, we're gonna put a G, and vice versa. C, G. Okay? So using that rule, let's go. If I see a T, I'm gonna put an A. I got A, U, G, A, C, C, U, G, G, A, G, U, C, A, C, G. Good. All right. After that, now this part may have confused you down here because it says to write the tRNA because the tRNA is actually attached to the amino acid, but I'll get there in a minute. So usually it doesn't ask you for the tRNA, so it just says translate, and then it says amino acids next to it. So you take this, you go down to the code wheel, AUG, A, you start in this quadrant, right? Because you start with the biggest letter. So we're in this quadrant down here now. A. Now we go to the next letter size. Here we go. U. Right, so now we're in this section. And then G is right here. And it says methionine there. But you just write the first three letters. M-E-T. And then it like makes a little peptide bond to the next amino acid. AAC. We're in this quadrant again. A. C is here. And actually, C and U both code for the same amino acid. It's called asparagine. Okay, it's right here. So we're going to write ASP. Just the first three letters of the amino acid. So this is interesting. So it says A, right? Sorry. AAC, right? So let's say there was a little mutation, and instead of AAC, we've got AAU. Well, that's still going to be asparagine, so that's called a silent mutation. It's not going to cause a difference in the protein. Okay, and then that's it. I'm not going over the rest. You should be able to get those by now. Let's go down to the bottom. All right, using the DNA template below, what would the complementary strand be? Okay, so here we have the template or original DNA strand, and then the complementary DNA, or the opposite side of the DNA, would just be the complement that we talked about before. So we're still doing DNA, so we're using T. We're not using U because it doesn't say RNA, so it's T, A, C, C, G, A, G, 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 T, T, C, A, C, T. Okay? Now we're making mRNA. We're making mRNA, and we're making mRNA from this strand. So that may have confused you. So if we're making mRNA from this strand, this is going to be, it depends on which way you did it. It doesn't really matter to me which way you did it. If you made mRNA from this strand, I'm okay with that. If you made it from this strand, I'm okay with that. Let's see, using the same DNA template, what would be the complementary? Okay, so complementary to this one, I think it's asking for. If you did this one, that's fine. So ATG, it's going to be UAC. Okay. And what you're going to see is that the mRNA is the same as the opposite side of the DNA, except the T's become U's, right? C, G, A, G, 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 U, U, G, A, C, U. Okay. This tRNA kind of interesting. tRNA is whatever's going to bond to this mRNA at the ribosome. Doesn't usually ask you to talk about tRNA, but I want to see if you could figure it out. So U, we're going to go back to A. A is going to be U, C is going to be G. Right? C is G, G is C, A is U. C, 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 and so on. Okay? The amino acid, it codes for, now this is the thing. If we go back up to here, you're thinking to yourself, okay, so do I use this to get my amino acid? Or do I use this? Right? Which one do I use? You actually are going to use this one. The mRNA wheel, it's always, these always have to do with the mRNA, not the tRNA. All right? So I would use UAC. And I would do UAC, and I would get tyrosine. So I would just put a TYR down here like this. Okay, and then just go across. You don't usually get questions about tRNA because it gets very confusing. This is called, remember, this is called the triplet. These three are called the triplet of DNA, 3 in a row. 3 in a row of mRNA is called the codon. And then these are actually called the anticodon. And this is the amino acid. So it goes triplet, codon, anticodon, amino acid. Really have to memorize those, so don't worry. Codon and triplet are the ones that are the most important. Okay, you don't really have to worry about the anticodon. But let's move on. Okay, I'm gonna put this one here because this video is getting a little bit long. I wanna make sure I can upload it for you. So I'm just gonna put this up and I'm not gonna go over it, but if you want to pause it and take a look at the notes I put on the side and what I underlined, check these answers, that would be good. I don't think these are that complicated. On the top of the page over here, I wrote at the top kind of what happens during protein synthesis. We go from DNA to mRNA to tRNA to amino acids, and then once those are all together, we get our protein. DNA to mRNA happens in the nucleus, that's transcription, and the rest of it is called translation. I put some notes on there that you could look over. Okay, and that's what we got. I'm gonna end this video here, and then my next video is going to be answering your questions about the genetic mutations work that I assigned this week, okay? Thanks, guys.